All right, here we go. So this is our next video on amino acid biosynthesis. I meant to click on things. And we're going to be looking at two particular types of reactions that are common in amino acid biosynthesis. These are also common in nucleotide biosynthesis as well. So the learning objectives, so after watching this video, you should be able to write the chemical equations for the biosynthesis of these four amino acids: alanine, aspartic acid, asparagine, and threonine. Describe the mechanism and kinetics of reactions catalyzed by transaminase enzymes. Predict the products of reactions catalyzed by transaminase enzymes. And describe the mechanism and kinetics of reactions catalyzed by amidotransferase enzymes. And then lastly, we're going to talk about this really cool thing called ammonia tunnels. So really we're going to be focusing on two enzymes, and they, like I said, they're common in biosynthesis pathways of molecules that contain nitrogen, like amino acids. All right, so again, the big picture here, last time we did nitrogen fixation, which is where you take the inert nitrogen from the atmosphere, convert it to reactive nitrogen, and then that could be used to make either GLU or GLN, and then GLU is used to make proline. Today we're going to be over here and here, and then finally in the last video we're going to get to serine, cysteine, and glycine. So again, like I said, we're going to be looking at these making these four amino acids: alanine, aspartic acid, ASN, asparagine, and threonine. So let's get started. The first enzyme we're going to look at catalyzes what's called a transamination reaction, and it's an enzyme that always has two substrates and always has two products, and we've actually met transaminase before several times. The overall reaction looks something like this. It's an amino acid plus an alpha ketoacid in equilibrium with an amino acid and an alpha ketoacid. And the reason that the substrates and products are so broad is because these enzymes that are called transaminases can have a variety of different types of amino acids as their substrates and a variety of different kinds of alpha keto acids as their substrates. We're going to keep it pretty focused on the products, where the products are either alanine as the amino acid, ASP as the amino acid, or GLU as the amino acid. Those are the primary transaminases that produce those three amino acids as the product. So let's get to drawing some of these. I'll pick a different color. Let's go blue. I'm going to move this up and out of the way. So let's draw some structures. I'm not going to draw the mechanism right now. We're going to do some chemical equations. So again, the overall equation is amino acid plus I'm going to do alpha Ka for alpha keto acid in equilibrium with amino acid plus alpha keto acid. So we're in nitrogen metabolism, which means we're really interested in the movement of nitrogens around. This is the basic structure of an amino acid. Coming off of a central carbon, there's an ammonia group, a carboxylic acid group, an alpha hydrogen, and then the R group. I'm just going to leave the R group as any amino acid. Here's our amino acid. And for this particular one, we're going to use the alpha keto acid that is pyruvate. So I'm going to put COO minus at the top to a carbonyl. Any molecule that has this connectivity of atoms, where it's carboxylic acid to carbonyl, is an alpha keto acid. For pyruvate, all we do is add a CH3 on the bottom, and we get pyruvate, which is an alpha keto acid because it has the connectivity of carbonyl to carboxylic acid. That's what an alpha-keto acid is. And all that's going to happen is the nitrogen on the amino acid, this group right here, is going to essentially swap with the carbonyl to create a new amino acid and a new alpha-keto acid. When we do this, the alpha-keto acid is going to become the new amino acid. So I'm going to leave my COO minus at the top. Excuse me, I gotta go back to my other, I'm in the highlighter mode. COO minus at the top, CH, NH plus, and because I'm using pyruvate as my alpha-keto acid, I know that there's a CH3 down here. And that, my friends, is ALA, or alanine. So we just made alanine, and what's left over is the alpha-keto acid. And I'm just going to put an R group here. So again, the nitrogen from the original alpha-keto acid, or the original amino acid, moves over to the alpha-keto acid. So it gets swapped over. I'm having a hard time with my highlighter here today. It gets swapped over and moves here. So the movement, the transaminase just moves a nitrogen off of amino acid to an alpha-keto acid to create a new one. And because this reaction uses pyruvate as its alpha-keto acid, and alanine is the product, it's a particular type of transaminase. I'll go back to this one. And we abbreviate it as ALT, which is the abbreviation for alanine transaminase. I didn't leave myself enough room here. I'm just going to zoom in really quick. So this is the abbreviation for alanine transaminase. Okay, so what that means is that this particular transaminase binds to pyruvate and creates alanine. And this transaminase is only found in the liver. It's going to become important later. All right, so we made alanine. Check. Let's make aspartic acid. So again, I'm going to go back. Let's play around with colors here. Doodly 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 doo. I'll leave my amino acid in blue. Or maybe I'll just do, yeah. What can I, okay. H3 plus, C, COO minus, H, oops, to an R group. There's my amino acid. Now, the alpha-keto acid I need in order to make aspartic acid is OAA. And I'm doing this in a particular order, so maybe I'll just do black. So again, alpha-keto acid is a carboxylic acid to the carbonyl. And now where pyruvate just had the one methyl group coming off of it, OAA has two. And then it's followed up with another carboxylic acid. So oxalo acid is an example of an alpha-keto acid. Oh, I did too many. Oh, goodness. Excuse me. Excuse me. Pyruvate is 3 carbons. OAA is 4 carbons. And OAA ends in a COO minus. So we've gone from an alpha-keto acid. We're going to just be growing the alpha-keto acids by 1 carbon at a time. So pyruvate is 1, 2, 3 carbons. This is OAA, which is also an example of an alpha-keto acid, and this has 1, 2, 3, 4 carbons. The products of this are going to be the new amino acid, which comes from the alpha-keto acid, so I'm going to leave it in black, and H3 plus, C, H, COO minus, and then I have my CH2 group, COO minus. And that, my friends, is aspartic acid or aspartate, plus the leftover alpha-keto acid, which I'm just going to abbreviate as R. So hopefully you're starting to see a little bit of a pattern. The alpha-keto acid is what gives rise to the new amino acid. The original amino acid gives rise to the new alpha-keto acid. That's how it always works. Other things that you might have noticed is there's always a COO minus at the very top of all the structures. You can draw it like that if it helps keep things very simple. The NH3 groups are kind of in this midline here. And then what's coming off of the carbon is either a hydrogen or a carbonyl, hydrogen, carbonyl. The R group is what changes between everything. All right, so that's one R group. Here's an R group, R group, R group. Food for thought. Okay, so we've made alanine. We've made aspartic acid. Oh, this enzyme that catalyzes this one is called AST. Oh. Which is aspartic acid transaminase. ASP transaminase. And this enzyme is found in lots of cells, like pretty much all cell types that you can think of. It's found in the liver, it's found in the heart, it's found in the brain, skeletal muscle, red blood cells. It's a very ubiquitous enzyme. Okay, so then we made ASP. So now all that's left over is to make glutamic acid. So let's keep marching through. I'm going to, again, do my amino acid in blue, NH3, to the chiral carbon. We'll call it the alpha carbon, carbonyl carbon, or the carboxylic acid, alpha hydrogen, R group. It's my amino acid. In order to make glutamic acid, we're going to need the alpha-keto acid that is called alpha-ketoglutarate. And alpha-ketoglutarate has 5 carbons. COO minus. That's how I know it's an alpha-keto acid. CH2, CH2, COO minus. And this is alpha-ketoglutarate, the common intermediate in the TCA cycle. Reaction proceeds, and I get my new amino acid, COO minus up at the top, C, NH3 plus, H, and then I'm just going to add these last 3 carbons onto the end of it, CH2, CH2, COO minus, and that is glutamic acid. And I am left over with an alpha-keto acid. Okay. Those are the transaminases. If you've noticed, all of these reactions are at equilibrium. And the delta G for all of these is pretty dang close to 0, which means that the directionality of the reaction is highly substrate dependent. So if you have a high concentration of alpha-ketoglutarate, then with Le Chatelier's principle, the reaction is going to push towards products and you'll get glutamic acid. If you had a high concentration of aspartic acid, then with Le Chatelier's principle, you're going to push more towards the reactants or the substrates, and you'll end up with more OAA. So that's just part of the functionality of the transaminases, is they're really, really dependent on whatever the substrate concentration is, and the substrate binding is the driving force for what's going to be favored in terms of products or reactants. Zoom myself back out here. Check. all the transaminases require a prosthetic group called PLP. And PLP stands for pyridoxal 5-phosphate. That is the structure shown on the left here. And the way that you get PLP is by eating vitamin B6. So you eat vitamin B6, which is called pyridoxine, and your body metabolizes it into PLP, and then from there, it attaches itself to a transaminase. And the reason that this is a prosthetic group is because it's covalently attached. To the enzyme. And then it helps the enzyme perform the reaction. So without PLP, the enzyme would not be able to perform the transaminase reaction. Other prosthetic groups that you have met before, let's see, lipoamide comes to mind in the PDC. Examples of what do we call things that aren't covalently attached that help the enzyme react? Things that are not covalently attached that help the enzyme do its function are called coenzymes. Coenzymes and cofactors also help the enzyme do its function, perform its reaction, but these are not covalently attached. That's the difference between a prosthetic group and a coenzyme or a cofactor, is how it associates with the protein. Prosthetic groups are covalently attached. Anyways, we just met another vitamin. Vitamin B6, pyridoxine, is the precursor to PLP, which is the cofactor, is the prosthetic group needed for transaminases to perform their reaction. The kinetics of transaminases is really cool. Transaminases follow what's called ping-pong kinetics. And I've tried to think about all the different ways that I can draw this out, and I think I'm going to do the Lineweaver-Burke plots first, and then sort of the schematic overview second. So ping-pong kinetics all follow a general sort of trend. So when we're going to look down this line, we're going to go left to right. Left being the first thing that happens, and then as you move to the right on the line is the second and the third and the fourth thing. So ping-pong kinetics start with this. Okay, so you have an enzyme. And the first thing that happens is the first substrate binds. And you get E and S bound together. And then you form the product, the first product, and the first product leaves. So substrate 1 comes in, product 1 is made, and then you're left with an altered E, and then S2 comes in, and you get enzyme that's been altered, is bound to S2, the reaction happens, and you get E, product 2, and product 2 then leaves. So the first substrate goes in, first substrate goes in, first product leaves. Second substrate comes in, second product leaves. That should have a little P2 on it. What this looks like in terms of the Lineweaver-Burke plots. Again, because we like to use the Lineweaver-Burke plots because they're easy visuals to be able to compare the different types of enzyme kinetics. So if you'll remember, we have 1 over V naught. This is going to be 1 over substrate 1 concentration. And this Lineweaver-Burke plot looks like this. None of the lines intersect, and this right here going down is in terms of increasing concentration of substrate 2. So what does this tell you? This tells you that as you increase substrate 2, you increase the Vmax. Well, that's a good thing, because then the enzyme is becoming more and more efficient, it's being able to spit out more and more product. What it tells you over here is that as you increase substrate 2, you're increasing Km. Km increases. Well, we know that that's actually not a good thing. But the reason that Km is increasing is because essentially substrate 1 and substrate 2 can compete with each other. So the more you increase substrate 2 concentration, the more it competes with substrate 1, and so the binding affinity goes down. This is called ping pong kinetics, and I'm going to draw the schematic so that you can see who's being pinged and who's being ponged. But the Lineweaver-Burke plot looks like this. They're 3 parallel lines that do not intersect. This probably looks very similar to a certain type of enzyme inhibition. But I'm going to go ahead and draw the schematic for you now so that you can maybe better see how ping pong kinetics works. So ping pong kinetics, again, is a type of kinetics where you have 2 substrates and 2 products, and the order of binding and product being made is very specific. So who's being pinged and who's being ponged? I'm going to just move this all kind of up and out of the way. So if you have like another piece of paper to draw on, this would be a good time to get another piece of paper. And I really like that pink color, so I'm probably going to stick with it. All right, so let's draw the enzyme, the transaminase. And the transaminase follows ping pong kinetics. So this Pac-Man is going to be my transaminase. I'll just put a T for, oh, maybe I shouldn't call it T. I don't know, let's call it transaminase. And the transaminase has a prosthetic group on it called PLP, so I'm going to do one single line, meaning a covalent line, PLP. So that is covalently attached. And what happens first is substrate 1 is going to bind, and substrate 1 is the amino acid, so it has the NH3 group on it. So substrate 1 is going to bind. It's going to pass the NH3 to PLP, and product 1 is going to leave, and that's going to be the new alpha-keto acid. So schematically, what is left over, how the enzyme is altered, is the PLP is holding on to the NH3 group. So in my mind, the NH3 group is the ball of ping pong, and the substrates and products are the paddles. I don't know what the enzyme is. I don't know if it's the net or what, but the NH3 to me is the ping pong ball, which is being passed back and forth between the paddles of the substrates and the products. So now substrate 2 comes in. S2 comes in, which is an alpha-keto acid, so I'm just going to put a carbonyl here. That's going to come in and take the NH3 group off of PLP, and you're left with product 2, which is our new amino acid, NH3+. Ping pong. And what you're left over with is the enzyme has been regenerated in that case. PLP. Yeah, and we could literally go backwards. So if we had product, let's just say we could go back to play ping pong again. P2 has the NH3 group. It could bind to PLP and spit us back out substrate 2, which is the alpha-keto acid. And then we would have the transaminase with the NH3 on it. And product 1, which was the alpha-keto acid, could come in and take this and get us all the way back to substrate 1, the original amino acid. So you can see how this enzyme is completely dependent on the concentration of the directionality of the reaction is dependent on the concentration of the substrates in the products. Anyways, in my mind, again, the ball is, I keep clicking on the eraser instead of the highlighter. Click here. This is the highlighter. Click on this. How about this and this? Okay, so the ball here is the NH3. It gets paddled to PLP, and then it gets paddled onto the product. So again, I see the paddles as the substrates in the products, and the ball as the NH3 group that gets ping-ponged around. Cool. That's the first reaction, and that gets us to 2 of our 4 amino acids. So again, this is our big picture image. I'm going to zoom in here because I'm going to edit on top of this. So we made we made alanine. And I'm just going to edit some of this image so that this now becomes a reversible reaction. And in order to make alanine, we need an amino acid coming in and an alpha keto acid coming out. We need the the prosthetic group, PLP. And the enzyme that catalyzes this reaction specifically is ALT, alanine transaminase. We've also made aspartic acid using a transaminase reaction. And in order to go from, again, the alpha keto acid to the amino acid, we need to input an amino acid and get out an alpha keto acid. This is a transaminase reaction, so it requires the prosthetic group PLP. And to make aspartic acid, that enzyme is AST. We did also make glutamic acid coming from alpha ketoglutarate. I'm going to just add it in down here because there's a lot of different ways to make glutamic acid. And you can use, instead of using the glutamic acid dehydrogenase that catalyzes this reaction, the GLUDH, there is a transaminase that will take alpha ketoglutarate into glutamic acid. Also requires PLP and also requires the input of an amino acid to get an alpha keto acid. Many ways to make glutamic acid. All right, those were our 3 transaminase reactions. We've got 2 more amino acids left to make, and we're going to do the aspartic acid to threonine really, really fast. So I'm going to do it just on this page. And this requires, there's 5 steps here. So there's 5 enzymes to get from aspartic acid all the way to threonine. I didn't actually include that many arrows. 1, 2, 3, 4. There we go. 5 steps. And it costs 2 ATPs. And it also costs 2 NADPH. If you remember, the NADPH is our coenzyme that is used to build fatty acids. It makes sense that it's also used to build amino acids, whereas NADH is used in catabolism, NADPH is used in anabolism. This is all I want you to know about getting from aspartic acid to threonine. The intermediate steps, I mean, if you want to look at them, you can. If you think they're fun, that's awesome. I'm not going to spend any time on it whatsoever. I like to do more bigger picture things and things that have more meaning and that we're going to see frequently. So that leaves us just with making ASN. And the way that we're going to make ASN, we're going to talk about it in a way that we talk similarly to how we talk about transaminases. And there's just a different enzyme that does that. And these enzymes are called amidotransferases. The similarities between these and transaminases is they also require at least 2 substrates. Dang, highlighter mode again. 2 substrates, at least. 2 products. So that's a similarity between amidotransferases and transaminases. They also can use a variety of substrates. So that's another similarity. So that's why we talk about these as classes of enzymes instead of talking about, things like we looked at hexokinase specifically and we looked at pyruvate dehydrogenase specifically. Transaminases and amidotransferases are more broad classes of enzymes because they catalyze a bunch of different reactions that are relevant to nitrogen metabolism. So let's take a look at the general reaction catalyzed by an amidotransferase, and then we'll look specifically at making ASN. I'm just going to move. What am I going to move? What am I going to do? Scoot. Scoot, scoot, scoot. I don't really need to scoot it that much. Okay. The amidotransferases have 2 main active sites. And we're going to look at these images going left to right. So this outline here is one active site, and this outline here is the second active site. And we call this first one on the left is called the glutaminase site. Okay. And then this second one here on the right is called synthetase. Okay. So what happens in each of these active sites? So in the synthetase, or in the glutaminase site, glutamine is bound specifically. So all of these amidotransferases use glutamine. What they use as their second substrate differs. So glutamine binds, and it binds to cysteine. And when it does that, when the cysteine reacts with the glutamine, it releases an NH2, which becomes NH3 ammonia. And this ammonia travels through a tunnel. We're going to talk about this tunnel in a minute because it's so cool. It travels through this tunnel to reach the second substrate. Now, the only requirements of the second substrate are that it either has to have an OH group on it or a carbonyl group. If it has an OH group on it, then you also have to use ATP so that it becomes activated. So then the NH3 travels through, and then depending on what your substrate is, it will either react with what was the alcohol, or it will react with what is the carbonyl, and your 2 products of those of the reaction are going to be glutamate, which comes from the glutamine, and then your other product. So again, two substrates in, 2 products out. One of the substrates is always GLN. The other one could be something, a substrate, a molecule that has an OH on it, or it has to have a carbonyl or a ketone group, R. And then your 2 products are always GLU plus whatever the other one is, but now it has the nitrogen on it. myself a whole lot of room at the bottom here. You get the idea. Again, if it's an alcohol group like this, then you also have to have ATP. So plus ATP, and then you'll get AMP, and pyrophosphate back out. We're going to look at a specific amidotransferase that makes asparagine. And we're going to talk about the tunnel. The tunnel is really, really cool. Okay, so the amidotransferase, which are usually abbreviated as GATs, glutamine amidotransferase, but then when they're specific to their substrate, they have totally different names. So the enzyme that we're going to look at that is a GAT, so this is a GAT, it's called asparagine synthetase, takes aspartic acid plus glutamine, and because it has an OH on it, we have to activate it with ATP, and we get the products glutamate out, asparagine out, and then AMP and PPI. So again, the enzyme that catalyzes this reaction, so we make ASN, is called asparagine synthetase. It's in the EC class, the enzyme class of GATs. And I found a couple diagrams to show this tunnel. So when the nitrogen comes off of glutamine, so when the ammonia comes off of glutamine, and the way that these enzymes are, pictures are drawn is the glutamine is on top, and then the receiving end piece is on the bottom. So hold on, let me edit. So ASP would be down here, and ATP would be down here, ASP. And they're showing AMP because that's one of the products, but we'll start with the reactants first. Okay, so GLN binds, and then the nitrogen, the ammonium, the NH3, gets ripped off of it, and it travels through a tunnel to get to the second active site. And they've done the tunnel here in this kind of like a chicken wire situation. And the reason there's a tunnel is because NH3 is toxic. You don't want NH3 getting out into the cell and wreaking havoc on the cell, and essentially is signaling apoptosis. So these enzymes that use or release NH3 have a way of trapping them within the enzyme so it doesn't leave. And the way they do this is through a tunnel. And I think that's so cool. This tunnel in asparagine synthetase is about 20 angstroms in length. So angstroms is a unit of length, a unit of distance. And it's not the longest. It's actually fairly short in terms of tunnels. So this is not the only enzyme that uses a tunnel, and it's not all enzymes use not all enzymes are tunneling ammonia. You could tunnel other things as well. It's fairly short in the scheme of tunnels. But the reason the tunnel is there is to keep the NH3 from seeping out into the environment. And I just think that that's wicked cool. And now we're going to talk a little bit about the kinetics. So just like with the transaminases, the amidotransferases also have two substrates and two products. And in the case of the arginine synthetase, I'm just going to lump the ATP in with one of the substrates you'll see, and the AMP and pyrophosphate in as lump it in with the ASN product. So I'm going to kind of do the similar thing with the order of events, because the amidotransferases follow the kinetics that are called ordered sequential, whereas the transaminases did the ping pong kinetics. The GAT enzymes follow ordered sequential kinetics. And this is the way that it looks doing that line diagram, and then I'll do the line mover Burke, and then we'll kind of wrap this thing up. All right, so you start with your enzyme, And then the first thing that happens in ordered sequential is substrate 1 binds, and you get the enzyme in substrate 1 is bound, and then you get substrate 2 bound. Now this is different than the ping pong because here you would have gotten the first product, but instead we get the 2 substrates bound, and then product 1 is released and product 2 is released. And then that gets you back to E. And we're going to talk about arginine synthetase, which is S1 and which is S2. But before I do that, again, I'm going to do the Lineweaver-Burke plot. Maybe I'll do a different color. Let's do blue. The Lineweaver-Burke plot. Like I say every time with the Lineweaver-Burke, the main reason that we look at Lineweaver-Burke plots is because visually, if you had a whole bunch of these lined up, you should easily be able to see, okay, that's ordered sequential. That is ping pong mechanism. That is competitive inhibition. That's mixed inhibition. That's uncompetitive inhibition. Things like that. It's a really easy visual. So in ordered sequential, it's 1 over velocity on the y-axis, 1 over substrate 1 on the x-axis, and all of, when you plot this, all of the lines, it's hard to do this, they all cross at a spot. All the lines cross. The defining feature of this graph is all the lines cross in the negative x, positive y plane. So again, if you remember ordered sequential, ping pong, none of the lines crossed at all. And then again, as we went down here, this was increasing substrate concentration. The same thing is happening in ordered sequential as we go down the lines. That is increasing substrate 2 concentration. And when you look at this, as you increase substrate 2, Vmax is increasing, and then Km is decreasing. All of those things are good, better. And for the specific case, so when we look at arginine synthetase, well, who's S1 and who's S2? Like, who binds first? Does glutamine bind first? Does aspartate bind first? Let's see, how do I do this? I'm just going to move this completely out of the way so I can draw big and not have to worry about it. So let me make my little enzyme active site. Okay, so I'll make this the synthetase to match before. The synthetase active site in glutaminase. All right, so for arginine or asparagine synthetase, it turns out that aspartic acid and ATP bind first. I'll just put them over here. I'll do I'm gonna put the H on this only to keep it consistent with the mechanism that was in the figure a few earlier, because the OH, because the aspartic acid, it's the OH that's going to be replaced with the nitrogen, so that OH has to be activated, so that's why I wanted to put that there. And then, so ASP and ATP, so I'm just going to put an A and then a P and a P and a P. Those two have to bind first, and that causes an allosteric change in the active site. So let's say this changes shape so that glutamine now binds and fits. glutamine, and the glutamine is still bound using that cysteine residue. H2, put the cysteine here, and again, attacks here, releases the N2, which becomes NH3, and that travels over here to the activated substrate. Let's see, activation of the substrate is going to attack here, cleave the pyrophosphate. We'll probably draw this out more in class. But the idea that I wanted to get at here is these two together in ordered sequential kinetics would be S1, and then glutamine is S2. So aspartate and ATP bind first. And that causes an allosteric change I'll just put delta allosteric change so that GLN can bind. And that makes GLN S2 in this case. All right, cool. So we did transaminases that follow ping pong kinetics and amidotransferases that follow ordered sequential kinetics. And then I just wanted to give a shout out that there are other enzymes that have tunnels. Not all of them tunnel ammonia. Some of them tunnel some other substrates. But I wanted to show you one other enzyme that we've met before. This is carbamoyl phosphate synthetase, so part in the urea cycle. And this tunnel, which you can see in the blue chicken wire, going, let's see, I can't even figure out what color is going to highlight on top of this. There's a lot of colors here. I guess I'll do pink. Going through here is an ammonium tunnel or ammonia tunnel. And this tunnel is 80 angstroms long. It's really, really long. I don't know why it's so long. It seems kind of silly. But there's it just has a really, really, really long tunnel for shuttling the substrates. Yeah, I just thought that was really cool. Anyway, and these enzymes with tunnels are really interesting. Not all of them tunnel ammonia. They can tunnel other substrates. In fact, if you're looking at this figure, you'll notice that ammonia is actually only tunneled until this site, and then the carbamate is tunneled all the way down to the second site. I'm thinking of these like water slides or something. I just think it's so cool. Shouldn't we do like a biochemistry themed like theme park, right? Where every ride is an enzyme or something, or I don't know. This one would be a water slide. Carbamoyl phosphate synthetase would be a water slide. You'd start at the top, you'd cruise on down, maybe you'd pick up a friend halfway through, and then you'd cruise on the rest of the way. Or maybe you'd pick up a snack. I don't know. You get the idea. That's all I'm going to talk about. I will see you when I see you.